I just want to thank everybody for joining us today for our Coffer Memorial Lecture. This is our second lecture to honor Dr. Daniel Ian Coffer. My name is Barbara Benlin, and I'm a professor in the Department of Medicine, and I lead the Wisconsin Alzheimer's Disease Research Center's Research Education component, which supports individuals who are doing research in Alzheimer's disease and related dementias. The Dr. Daniel Ian Coffer Lecture is an educational program devoted to shaping future generations of dementia care professionals. This is our second annual lecture, and we're indebted to the Coffer family for making this lecture possible. Dr. Coffer was a talented neurologist specializing in frontotemporal dementia and Lewy body dementia at the University of North Carolina at Chapel Hill. And he started his journey here at the University of Wisconsin, Madison, and received his medical degree here. He was a founding director of the UNC Memory Disorders Program and chief of the Memory and Cognitive Disorders Division in the Department of Neurology. And this year, we are very honored to have Dr. Roberta Diaz Brinton as our keynote speaker. Dr. Brinton is the director of the Center for Innovation in Brain Science and a professor in the departments of Pharmacology and Neurology at the University of Arizona. And she will be speaking today on the topic of regenerating the Alzheimer's brain, Allopregnanolone as a regenerative therapeutic proof of concept from discovery to clinical trial. And at the end of this presentation, you'll have the opportunity to ask Dr. Brinton questions. So at this time, I would like to turn this over to David Coffer, who is the brother of Daniel, to share a bit about Dr. Coffer and this series. David, please.
Thank you, Barbara. Good afternoon. Our brother and partner, Daniel Ian Coffer, believed in a humane neurology that combined the promise of research with the challenge of present-day realities. Dan prioritized the dignity and well-being of his patients, and he believed in empowering caregivers with knowledge and training rather than sidelining and leaving them on their own. UW was Dan's beloved alma mater, and when almost 2 years ago, he faced an unforgiving cancer, and when he thought about the best way to inspire new generations of neurologists and degenerative scientists in his values, he endowed this lecture series as one of his last acts. Our family is grateful and humbled to have the opportunity to participate in the second Daniel Ian Coffer lecture with the esteemed Dr. Brinton. Thank you very much.
Thank you very much, David. I'd now like to introduce Dr. Sanjay Asthana, who is the Associate Dean for Gerontology and Professor and Head of the Division of Geriatrics and Gerontology, as well as the Director of the Wisconsin Alzheimer's Disease Research Center, and he will be introducing Dr. Brinton.
Thank you so much, Dr. Benlin. Good afternoon, everyone. Welcome to the series. It's truly a pleasure and a privilege for me to introduce our keynote speaker, Dr. Roberta Diaz Brinton. Dr. Brinton and I have known each other for over 20 years, and we shared some common research interests. So it's really a privilege to hear from her and be a part of this such a critical series. Dr. Brinton is a world-renowned scientist and an expert in Alzheimer's research. Particularly, she focuses on mechanisms that underlie the increased risk for the disease in the female brain. And for both men and women, Dr. Brinton is developing one of the first regenerative therapies for Alzheimer's disease. She's the inaugural director of the Center for Innovation in Brain Sciences and professor of pharmacology, neuroscience, and neurology at University of Arizona. The Center for Innovation in Brain Science is a hybrid university biotech research environment with a focus on therapeutic development for neurodegenerative diseases, including Alzheimer's disease, Parkinson's disease, ALS, etc. Dr. Brinton's research spans discovery, translation, clinical science, and she has published numerous renowned high-profile papers that are really the leading aspects of Alzheimer's and hormone treatment research. In addition to her research endeavors, Dr. Brinton has trained a number of trainees right all across the levels of training. These learners are at the undergraduate, graduate, postdoctoral level, faculty level. And also she has established a program that involves a STEM education program that involves over 600 inner city high school and elementary students in Los Angeles. And she's also mentoring currently a number of Latinx and American Indian students. Dr. Brinton is the scientific, her research accomplishments really have received a number of accolades, and among these awards include the National Institute on Aging Merit Award, the Scientist of the Year Award by the Alzheimer's Drug Development Foundation, the Goods Prize for Excellence in Alzheimer's Drug Discovery. She has been described as the 10 best minds by US News and World Report, and received the US Presidential Citizens Medal from President Obama. She's also on the National Institutes of Health Directors Advisory Committee, the National Institute on Aging Scientific Advisory Committee, and the Alzheimer's Drug Discovery Foundation Board of Governors. So it truly is a privilege and an honor for me to invite Dr. Brinton to make a presentation. Dr. Brinton.
Well, thank you, Dr. Astana and Brenlin, for that introduction. And especially many thanks to the Coffer family, and particularly to Daniel, who I am honored to represent today. I will share my screen and start my talk. All right, so today I want to describe and share with you a journey that has been quite some time in the making, and that is our goal to regenerate the Alzheimer's brain and the development of Allopregnanolone as a regenerative therapeutic from discovery to clinical trial. And I'm going to get the laser pointer here and go forward here. These are my disclosures, and I'm going to close this. These are my disclosures, our sponsors, patents relevant to the work that I'll be presenting today, and our development of a startup, New Therapeutics. I want to first acknowledge that this is team science, takes a village, literally a village. And these are the people that have been involved in this journey from discovery to clinical trial. And I particularly want to acknowledge the clinical trial participants and their caregivers who have been instrumental in bringing the first steps of our regenerative therapeutic to life. So the challenge, as you all are likely aware, is that Alzheimer's disease is a progressive neurodegenerative disease. There are multiple steps in this entire pathway for the development of the disease. And one of the first aspects of the disease is actually a decline in the regenerative capacity of the brain. And this is the usual therapeutic intervention time where there's already beta amyloid plaque, there's already white matter degeneration, there's already neurofibrillary tangles. And we're really targeting an earlier stage of the disease where individuals have been diagnosed with mild or early Alzheimer's disease. Hopefully we can intervene at this point when the brain still has regenerative capacity. So as I mentioned, we know a great deal about how rapidly the brain degenerates. Hopefully I can get this going. And what we don't know a great deal is how rapidly the brain can regenerate. And that's obviously our goal, is to regenerate that degenerated brain. We are targeting 2 regions of the brain that have regenerative capacity, and most our target is primarily to the hippocampus, that area of the brain that you're utilizing extensively right now. And I want to just make sure I can see this. And sorry. The area of the brain that you're using quite extensively right now to encode new information and hook up that new information to your existing body of knowledge. And let's make sure, there we go. There, finally. So this gives you a diagram, a video of these regenerative zones in the brain. And the one that we're most focused on here is the hippocampus, because it's so critical for learning and encoding new information. And the hippocampus has a zone called the subventricular zone, where new neural stem cells can now regenerate and repopulate that area of the brain, the hippocampus, that is critical for learning new information like I hope I will be presenting to you today. This just gives you a snapshot of many decades of work that has gone into bringing us forward from our discovery and understanding of the mechanisms of action, our translational research and development, our clinical trials, regulatory strategy, and now formulation development. So I'll take you first through our discovery science, a few snapshots of our discovery science. And what we discovered is that allopregnanolone can promote the generation, neurogenesis, in a mouse model of Alzheimer's disease. And you see those newly generated cells here in green. We also determined that allopregnanolone, or ALLO for short, ALLO, ALLO can promote the regeneration of human neural stem cells derived from the human brain. And what we see here is the dose response curve. And this is going to be very important as we go forward, because that dose response curve obviously tells us the optimal dose, but it also tells us that ALLO is designed for safety. It has been designed and selected for both efficacy and safety. So what this tells us right here is that more does not get you more proliferation of these neural stem cells. It actually gets you less. And that's critically important. And what work from Spalding et al. Showed a number of years ago is that the aging brain is still capable of regeneration, and about 700 new neurons per day. Again, important for our therapeutic target audience. So what in the triple transgenic mouse model, what we were able to see is that in the normal animals, there was a small effect, but not significant. Whereas in the Alzheimer's mouse model, there was a significant increase in neurogenesis. And again, what we find throughout our preclinical and clinical research is that ALLO restores normal regeneration. It does not exceed normal. As I mentioned, there are two regenerative zones. Here is the subventricular zone. I'll be talking mostly about the subgranular zone, but here in the cerebral cortex, there's also a regenerative zone. And we see that in ALLO-treated Alzheimer's mice, there is, again, a significant increase in the generation of these neural stem cells, which was very encouraging, that in the two brain regions capable of regeneration in the adult brain, we could see that ALLO promoted regeneration in both of those. Along the way, we also discovered, we weren't necessarily looking for this, but it was another serendipitous discovery. And what we found is that ALLO, here what you see is beta amyloid plaques in this mouse model of Alzheimer's disease that increases with age, and that ALLO could significantly reduce two factors of the disease, two hallmarks of the disease, beta amyloid plaque generation, as well as phosphorylated tau, which is critical to the formation of the neurofibrillary tangles. So in addition to promoting the regeneration of neural stem cells and neurons in the brain, allo was also reducing the burden of pathology. And that's critical because generating new neurons while the rest of the disease continues is likely not to be effective. Whereas if we have a therapeutic that both promotes regeneration and reduces the burden of disease and the mechanisms driving the disease, that will be obviously of greater benefit. We have worked through the mechanisms by which allo promotes neurogenesis, regeneration, and it's a wonderful mechanism because it both promotes regeneration while also having multiple safety factors. So here, allo activates a channel, the GABA chloride channel complex, which in mature neurons leads to a reduction in the excitability of the brain. However, in neural stem cells, because of a special channel that I'll talk about more in a moment, the intracellular concentration of chloride is quite high. So now when allo opens up this GABA chloride channel complex, chloride moves from the intracellular space to the extracellular space. And that's important because then that depolarizes the membrane, the neural stem cell membrane, and activates what is an L-type calcium channel that allows calcium to now enter into this cell and activate this entire regenerative pathway. And I'll come back to that calcium component because as it turns out, it's very, very important. So you have to, in this case, for there to be proliferation or regeneration of neural stem cells in the brain, you have to have this GABA chloride channel complex, but you also, I'm sorry, this is a little annoying. You have to have a special channel, the NKCC1 channel, and the membrane has to be depolarized, and there has to be this calcium channel. Again, all of these multiple factors that must be present that build in the safety of allo. Now, what we discovered also is that this can happen very rapidly. So allo was infused IV, and we could detect the first signal in this pathway, this phosphorylation of this this transcription factor within 5 minutes, then was followed by about 4 hours. We could see the activation of a neuronal differentiation transcription factor, and ultimately 24 hours later, measure neurogenesis, a marker of neurogenesis. Now, when we ask the question, does this have a cognitive impact? We're generating new neurons, we're reducing the burden of pathology. We then went on to ask, is there an impact on cognitive behavior? And what we were able to detect, is that indeed there is. And we really put these new neurons to the test. So we treated mice, these Alzheimer's mice, with allopregnanolone and a chemical BRDU that would label newly generated neurons. And we treated them for once at the beginning of the month, and then we put them back in their cage. And we then waited 7 days before we brought these animals back out, and we began to put them through an associative learning task. So they had to associate a tone, an auditory tone, with a air puff that would generate a response in these animals. And what we found is that the animals could actually learn this task. That's great. Then we put them back in their cage, and we waited another week. And then we brought them back out and we said, how well do you remember what you learned last week? And what we found is that in the vehicle-treated animals, those animals they were performing at chance. So they hadn't learned, and they certainly hadn't remembered. Whereas the animals that were treated with allopregnanolone relative to normal animals that were not Alzheimer's animals, allopregnanolone completely reversed that learning deficit. So the allopregnanolone treated animals behaved like normal. And these animals then, we then looked at how well their brains had generated new neurons. And we found that indeed that the allo-treated animals had indeed generated a substantial number of neurons that had survived the entire month, suggesting, very indicative, that those newly generated neurons that are actually designed specifically for this associative learning task type of function, associating new information and remembering that information. Okay, so from cognitive perspective, we showed that there was a significant improvement and restoration to normal. Overall, many years of research has shown that I'm really putting you through your paces here. Allo is promoting neurogenesis as well as the generation of both neurons, gray matter and white matter, and newly generated synapses are all being generated. We also have found that allo is promoting the bioenergetic system of the brain, and I'll talk about that in a moment, as well as reducing the inflammatory component. And I showed you the data where there's a reduction in Alzheimer's pathology. And we think that's largely through both promoting the bioenergetic system of the brain, as well as promoting cholesterol efflux out of the brain. So this is work that we've not yet published, but I'm very excited about. And that is what we've found is that in these neural stem cells, allo promotes the mitochondrial function, that actually the generation of energy, ATP, in these mice, and does so through this calcium signaling pathway that I described earlier. Now, we've also found that in astrocytes, another cell in the brain, part of the glial cells in the brain, what we find is that allo promotes the generation of new mitochondria. And those mitochondria essentially, again, increase the energy capacity of the brain. So both in neural stem cells, as well as in astrocytes, allo is promoting the bioenergetic system of the brain. First, in neurons, by promoting mitochondrial function, whereas in astrocytes, it's promoting the generation of more mitochondria. And all of that leads to an increase in the ability of the brain cells to generate energy and ATP. And we find that it reduces reactive oxygen species, free radicals, as well as reducing inflammation in the brain. And why this was a conundrum to us, because allo does not bind to a nuclear receptor, right? So it doesn't go into the nucleus and activate a whole bunch of genes, as estrogen and progesterone and testosterone do. However, by acting through the calcium signaling pathway, as I showed you for neurogenesis, it's through this calcium signaling pathway that then, through Krebs in neurons and this transcriptional regulator, PGC1 alpha and NRF1, through these calcium activated neural transcription factors, we can see this panoply of systems of biology being activated by allopregnanolone. Okay, so one of the questions that we really addressed was, what's the optimal treatment regimen? So what we found is that, as I showed you, I'm going to go to the laser pointer. So it stops. So what we found is that if we treated once a month, we could promote neurogenesis very well. What it didn't do is have an impact on the burden of pathology. If we treated 3 times per week every other day, we could reduce the burden of pathology, but we didn't promote neurogenesis. Again, that dose response curve for safety. However, what we discovered is that if we were treating once per week, we could activate neurogenesis, allow those cells to move out of the regenerative zone, and then we could reactivate that system. And through that paradigm, we had both the ability to promote neurogenesis as well as the reduced risk or the reduced burden of pathology. And so that was the optimal treatment regimen that we have gone forward with in our discovery research, as well as in our clinical research. So we know that the brain ultimately loses regenerative capacity. So we knew that we'd have to go into a population that was earlier in the disease process. So in our phase 1 clinical trial, we targeted individuals who had mild cognitive impairment due to Alzheimer's disease or early Alzheimer's disease, with the idea that this would ultimately be our therapeutic target population. So Allopregnanolone is a neurosteroid that we all make during our lifetime, and that declines with age. The highest level of allopregnanolone is actually induced during the 3rd trimester of pregnancy, when the brain undergoes a 4-fold increase in size. And why that's particularly important is obviously because we think this has the potential to impact, obviously, regeneration. So Allo has undergone the most challenging safety test there is. It's safe in women of reproductive age with fetus, both male and female. So thank you, Mother Nature, and all the mothers who have promoted neurogenesis in utero. There is an extensive body of data, both from preclinical work as well as in humans, that allo is safe. Now, the FDA required us to conduct 2 safety studies in 2 species and both sexes. We treated rats for 24 weeks of exposure and dogs for 9-month chronic exposure using our once per week. And essentially from this work, we found that there was no toxicological relevant changes in body weight, food consumption, or organ integrity. So following chronic exposure of once per week allo has proven to be safe. So this is the summary of our design of our phase 1 clinical trial. We conducted a phase 1 clinical trial in both women and men, 50% female, 50% male, in individuals who had an MMSE score greater than 20. So MCI due to Alzheimer's or early AD from 55 to 85 years of age, with the average age being 76 years of age, and 12 postmenopausal women and 12 age-matched men. This was a double-blind, placebo-controlled phase 1b/2a clinical trial. Our primary outcome was safety. We also investigated conducted MRI to establish whether there was any amyloid-related imaging abnormalities, as there are with the monoclonal antibodies like Aduhelm. And we were very fortunate to fall within this time zone when, if you'd shown that your molecule had any impact on beta amyloid, you had to conduct these MRIs for safety. And we conducted a number of exploratory outcomes, which, because the FDA required us to do the MRIs, we were able to really asked the question, which I know we were quite surprised at the outcomes, whether MRI could serve as this surrogate biomarker of regeneration. We also conducted cognition and conducted analyses to determine whether we could have predictive responders, a predictive outcome for responders. And again, we used our self-renewal regenerative therapeutic paradigm. Again, they were equal. We recruited for equal numbers of women and men. 62% of our participants carried the ApoE4 gene, 4% were 4/4s, and the remainder were 3/4 carriers, and 38% did not carry the Alzheimer's risk factor gene ApoE4. They were primarily non-Hispanic whites. We did have Hispanic and Black participants. So outcomes of the trial indicated that there were no indicators of allo-related toxicity, no evidence of microhemorrhages or amyloid-related imaging abnormalities. There were predictive pharmacokinetics. There was a sex difference in the maximally tolerated dose, with men being more sensitive to allo than women. However, there was no sex difference in the maximally effective dose. We established that cognitive analyses were feasible in this population, that the potential of using MRI as a surrogate biomarker of regeneration was feasible, and that the inducible pluripotent stem cell could serve as a predictive biomarker of responders. And the full analysis of the primary outcomes can be found here or using that QR code. So now to our exploratory outcomes. So if allopregnanolone is promoting neurogenesis, could we detect an impact on brain structure? And this just gives you the paradigms that we use to conduct those analyses. And I can talk more about that, but essentially, we've been very fortunate. Adam Rakes has joined our group and conducted these very rigorous MRI-based analyses to determine hippocampal volume, as well as changes in white matter integrity and volume. Okay, so at baseline, there was no difference essentially between the in either at baseline before any treatment between placebo and the allo targeted group, and also in the ApoE4. So there was no significant difference at baseline in either of these groups for the hippocampal volume. In the left hippocampal volume following treatment, what we found is that there were multiple responders in the allo group that showed a modest increase or stabilization or increase in left hippocampal volume. And that there was no difference between females or males. Both were responders, 3 of the males and 2 of the females. What we did find, however, is that there was an ApoE genotype effect, that allo essentially was promoting the potential for regeneration only in the ApoE4 carriers. And that the optimal dose was the 4 milligram dose. Out of the 3 doses that we tested, the 4 milligram dose was the optimal dose. Now, what's very interesting about the 4 milligram dose is it generates a blood concentration of about 152 nanomolar, which exactly matches the 3rd trimester of pregnancy, which was very interesting to us. So this was for the left hippocampus. Now in the right hippocampus, we saw the same pattern where allo promoted either stabilization or a slight increase in hippocampal volume, with 12 weekly treatments in both females and males. And that again, it was selective for ApoE4 carriers, and again, selective for the 4 milligram dose of allo. So we then went on to ask if allo is promoting the generation of new white matter cells, myelin cells, generating cells, oligodendrocytes, then changes in white matter structure and integrity should be evident. And to do that, we conducted fractional anoscopy, which is an indicator of white matter integrity, higher values are better, and a quantitative anoscopy, which is an indicator of the connectivity of these white matter tracks. And again, we conducted analyses to determine change from baseline. In this case, the red fibers indicate the left to right white matter tracks. Blue is superior to inferior from top to bottom, and green is from anterior, from the back, from the front of your brain to the back of your brain. And what we found is that the change in white matter integrity was significantly increased in individuals who were treated with allo. And this was in about 690 tracks, whereas in the placebo group, what we found is that that white matter integrity was decreasing over that same period of time. In the case of quantitative anoscopy, what we found, again, in almost 1,500 tracks, that allopregnanolone promoted the connectivity of these white matter tracks in a significant number of those tracks, and that's shown here. Either increased it or maintained those tracks. So there was a significant reduction in the loss of that connectivity. So then we began to ask, well, what is being connected? Oops. And the connectivity, when we looked at the connectivity across these networks, what we found is that there was significant differences and significant increased connectivity in the allo-treated individuals. And then we went on to ask, what are those fiber tracks? What are networks that are being significantly impacted? And what we found is that there was a significant increase in those hippocampal, that limbic area of the brain, the networks in the limbic area of the brain, the area of the brain that you're using most extensively right now, and the cerebral cortex, the sensory motor cortex. In addition, there was a significant increase between the connections between the limbic system, again, that area that you're using right now, and the default mode. And that default mode network is quite important to sustaining the functionality of the hippocampus and this whole area of the brain required for cognition. And we did see that there was a decline in the cerebellar and subcortical networks. And that's likely because of the remodeling of these synaptic or these white matter tracks. Because in Alzheimer's disease, what we know is that these cerebellar and subcortical networks try to take over some of that cognitive function that is being done by the limbic system and through the default mode. So what we find is that these are being promoted, whereas these auxiliary or backup systems are now being slightly dismantled. Okay, so as we've shown that the allopregnanolone is promoting white matter integrity and functional connectivity. We then asked, can we identify people who are potential responders like they do with cancer, right? So can we take a cell from that individual and then ask, are you a responder? Will you be a responder to this therapy? And essentially what we found is that, yes, when we looked at the plasma of these individuals who were treated with allo, we found that there was a reduction in these inflammatory signals, inflammatory cytokines. And then there was also an increase in the immune cells or immune signaling molecules that are involved in tissue repair and remodeling and healing. So that was quite exciting. We then also went on to do a series of analyses using what's called inducible pluripotent stem cells. So what we did was withdraw blood from individuals prior to being randomized, once they'd been accepted into the trial, and prior to the time of being randomized into placebo or treatment, we drew blood from those individuals and then took that blood back to the lab and took these peripheral mononuclear cells and reprogrammed them into what's called inducible pluripotent stem cells. And then we differentiated those cells back to neural stem cells. Okay, and then we asked, blind to whatever treatment they were on, whether those cells responded to allopregnanolone. And what we found is that in the entire cohort, actually in dose, the first and second doses, regrettably, we weren't able to do this on the third dose cohort, but on the first two dose cohorts, we were able to see that there was a positive correlation between allo induction of mitochondrial function, ATP generation, and an increase in left hippocampal volume. When we then asked the same question in only those individuals who carried the risk factor, and that we now knew from the brain analysis, the brain structure analyses were the responders, what we found is that there was indeed a very high correlation, 0.71, between those individuals who were treated with allo and their ability to regenerate the left hippocampus. So that's the good news. The challenging news is that this is a very long process. It takes months to do this. This whole process of gaining the cells, reprogramming them, and then differentiating them into neural stem cells. So unless that process becomes more rapid, can be increased, it's unlikely that this is a process that would be helpful, but it was very, very important to us that in fact we could see that there is the potential for a peripheral biomarker to identify responders versus non-responders. So we're continuing with that and hopefully increasing or decreasing the time that it takes for this whole process. So going forward, we have now designed a phase 2 clinical trial that will begin recruiting, the Regen Brain trial that will begin recruiting in likely January of 2023. In which we will enroll 200 individuals and randomly assign them to either treatment of allopregnanolone or placebo for 12 months. We will then undergo a crossover design where these placebo individuals will receive allopregnanolone for 6 months, as I know we're very excited about. So our primary analyses will be conducted at 12 months. And our primary outcome, and we're really putting it to the test here, our primary outcome will be change in hippocampal volume. Can we actually establish that there is, instead of loss of hippocampal volume, either stabilization or regeneration of the hippocampus? We know that atrophy and loss of hippocampal volume correlates incredibly well with decline in cognitive function and burden of pathology. So we're now asking the reverse question. Can we actually determine whether there is an increase in the hippocampal volume? over the course of 12 months. Our secondary outcomes will explore the impact on cognition, obviously safety and tolerability. We have multiple biomarkers that we will be exploring, again, with the potential of identifying potential responders or non-responders, clinical outcomes, and many different imaging outcomes beyond hippocampal volume. So this is where we are now. We have extensive discovery and translational science that supports our clinical science over multiple decades now. We've conducted a phase 1 safety and maximally tolerated dose clinical trial and a double-blind placebo-controlled trial that is suggestive that there's regenerative potential in the allo-treated group. We've completed our FDA IND enabling chronic toxicology. We are about to begin recruiting in our phase 2 clinical trial for proof of concept, and it'll be a 20 multi-site clinical trial in 4 milligrams over 78 weeks. And we're now developing multiple strategies for administering allo. Currently, this requires intravenous infusion once per week. And we're looking for strategies, we're developing strategies where there can either be in-home nurse care or self-administration or administration by a caregiver. And with that, I will thank again all of the team members who contributed to this work, and grateful thanks to our sponsors, particularly the National Institute on Aging, Alzheimer's Drug Discovery, and the Alzheimer's Association, and our collaborators. And with that, I will thank you and take questions.
Thank you, Dr. Brinton, for a wonderful presentation. If audience members have questions, please feel free to drop them in the chat, and we'll pose your questions to Dr. Brinton. I would like to start off with one, and I'm just wondering if you could talk a little bit more generally about how, changes in hormonal levels at menopause affect risk for Alzheimer's disease.
Well, we know that in the female, well, so let me just back up a step and say, most of our work has been in the studying the aging female brain. However, outcomes of that research has really taught us the right questions to ask of men. And a couple of take-home messages for that research, take-home points. And the first is midlife aging is a critical period, a critical window for setting up risk for later life development of Alzheimer's disease. And the menopausal transition has been very informative in terms of understanding that risk trajectory. And those women who are, what we found is that estrogen promotes and activates the bioenergetic system of the brain, literally from glucose uptake to glucose metabolism to conversion to ATP energy. It's also an anti-inflammatory. And when there's a decline in estrogen in the brain, can be a decline of about 20 to 25% in glucose utilization. That activates a starvation response, and the brain sends out a signal that indicates, I'm starving, we need auxiliary fuels. Good news, bad news. The good news is the brain is sending out this, I need auxiliary fuels, and is adapting. The bad news is it can use lipids as its fuel source, and the brain is the most lipidated organ of the body. And what we find is that the female, this is not true for all females, but vulnerable females can actually catabolize their own white matter for fuel. And that is likely what we find in this menopausal transition is that it's the transition state. It's just like puberty. This is a complex process that occurs in steps. and can expand a number of years. And that the symptoms of menopause are typically neurological. And that's really telling you that the brain is undergoing a significant restructuring and reordering. So for women who have increased inflammation and multiple severe symptoms of menopause are most likely at risk. And in addition, who lose control, metabolic control of glucose homeostasis. Now, what about men? Well, there's an abundance of research now that shows that the loss of testosterone can have multiple effects that mirror that loss of estrogen. Loss of testosterone, for example, can induce hot flashes, as one example. We published a paper, several papers recently, that hormone therapy can reduce the risk of developing Alzheimer's disease when given during the menopausal transition, when women are symptomatic, not later. We also published a paper, report that, regrettably, well, let me finish with the women, and that breast cancer therapies, particularly the aromatase inhibitors, which was surprising to me, as well as tamoxifen, can also reduce the risk of developing Alzheimer's disease later in life. Not to the same extent that estrogen, it's almost half, so it's a 25% reduction versus a 50-60% reduction. Regrettably, for men who are on anti-androgen therapies for prostate cancer, there actually is an increased risk. So anti-androgen therapies have a risk for multiple age-associated neurodegenerative diseases. So avoid prostate cancer at all costs.
Thanks for that really comprehensive answer. And I know that there's a lot more to know and learn on that. So I appreciate your very clear response on that. Do we have other questions from the audience?
Hey, Robbie, just a brief question for you. So what an outstanding presentation. And you really demonstrated how multi-step approach is so critical to addressing such a complex area. I know that your focus is on hippocampal volume, which is so appropriate in the disease we are targeting. I wonder if, and I've no doubt in your phase 2 trial, you'll be collecting all kinds of 80 biomarkers, Alzheimer's biomarkers, blood, CSF, and so on and so forth. Is there some understanding what you might predict in terms of effects of LO on the proteinopathies of Alzheimer's and some other neurobiological mechanisms that mediate that pathology? I know you'll get some information from phase 2 trial, but I wonder if there's some speculation in that area, how it affects the Alzheimer's neurobiology itself.
Well, I really think, Sanjay, it's really around the bioenergetic system and the inflammatory system. Now they go hand in hand. There's not this metabolic change and no immune system change. They're actually this complex interaction. And so the fact that allo can promote mitochondrial function as well as an anti-inflammatory, I think is very, very important. The other aspect of this is when we're looking at these biomarkers, obviously I think it's going to be important to look at the new biomarkers for both beta amyloid and for tau and the phosphorylated tau. So we're going to be looking very closely at that. I will tell you that we didn't plan for that initially. So we didn't have sufficient number of samples that fit the criteria to do that from our phase 1. But we certainly will be doing that going forward.
Yeah, no, that's so critical. So from animal research, has someone looked at the effects of LO on amyloid plaque formation or tangle formation at all?
Well, yeah, so we didn't look at tangle. We did look at beta amyloid generation.
Okay.
And saw that allo reduced beta amyloid load in the brain, as well as phosphorylated tau, while at the same time increasing myelin basic protein and another marker of white matter. So neurogenesis, markers of white matter were increased, while beta amyloid, inflammation, and phosphorylated tau were decreased. And so part of something that I became frustrated with was, how is it that allo can induce this multiplicity of outcomes, and we don't have any evidence that allo binds to a nuclear receptor, right? And that's why we were so thrilled, and it's been several years in the making, but to now establish that allo is working through this calcium signaling pathway, and it's the calcium signaling pathway that is activating these transcription factors, both for neurogenesis as well as essentially reducing the burden of pathology and promoting synaptogenesis and cognitive function. It's like, hallelujah, we finally have a common mechanism.
Yeah, you got the magic compound. It's acting at so many steps, which is just so impressive.
And I think it's a very important point, Sanjay, because of the complexity of Alzheimer's disease, I think we are going to require a systems biology approach.
Absolutely.
That simply removing either beta amyloid or phosphorylated tau is not going to be sufficient, because those are the downstream consequences of upstream systems of biology. And we really have to move upstream to ask what, as you just did, what are the mechanistic drivers of Alzheimer's disease? Not what's the outcomes, but what are the drivers, so that we can begin to change that trajectory.
Absolutely. Yeah, thank you, Robbie.
Target the cause.
Dr. Brinton, you mentioned that allo increases during pregnancy. Has anyone done imaging of imaging of the brain during pregnancy to see if those peaks in allo maybe have an impact?
Well, we know that the brain increases fourfold during the third trimester of pregnancy when allo is high. In animal models, if allo is not present, then there is a dramatic impact on neural development. My colleague, Dr. Naila Zaghul, is looking at the regenerative capacity of allo in, and this is in animal studies, in neonates that have undergone ischemia, as what occurs in cerebral palsy, and what's the impact. And it's really quite astonishing. I'm not going to steal her publication thunder, but it's just very, very exciting, and we're pretty excited about it.
Wonderful. What about in the women that are pregnant?
So I think it's a great idea. It's a great question. What happens to the female brain? And what I think is, I honestly think a lot of the sedation that women can experience during the third trimester of pregnancy is in part due to, obviously, you have this energetic demand, so you're tired, you're definitely tired. So whether allo is contributing to kind of a sedative effect, it certainly could be promoting anti-anxiety. It's like, I'm too tired. I'm, obviously being facetious, but I think it's a great idea to actually image women pre, during, and post.
Yeah, it would be fascinating. Any other questions? Well, we're getting some accolades. I know this was a really fascinating lecture for us. We had a number of individuals attend. I want to thank you, Dr. Brinton, for joining us today and sharing your really exciting and promising work that is really that I believe is going to help a lot of people. We really appreciate hearing about this trajectory of work. I also would like to thank David, Nancy, and Denise for joining us today, and thank your entire family for making this lecture possible. And thank you everybody for joining us. And we look forward to seeing you again next year for our Daniel Ian Coffer lecture.
Thank you again.
Thank you, Robbie. All the best.
Thank you. It's you too.
Yeah.
That
Bye.
little halo in the back, I think, is Daniel.
Right.
Thank you.
Bye.
Bye.
Thank you, everyone. Bye.